Hi folks, so this is chapter 14, the brain and cranial nerves. Uh, just so you know how the chapter unfolds is the beginning of the chapter introduces you to the brain and then comes back to the brain for some more details. So you'll get an overview of the brain. And I'm going to try to resist going into all the details of the first few brain pictures you see because then towards the end of the chapter, we come back to the brain and start talking about some of the details. So I'll try to resist that. And then the very end of the chapter, the last section, is the cranial nerves, which is mainly lab stuff. However, we'll talk about it a little bit in lecture. So let's get started. So there's 6 regions to the brain. Those 6 regions are, as you can see there, the cerebrum. The cerebrum has 2 layers: the cerebral cortex, the gray matter, and then this white matter, which projection fibers exist in. And it's, by the way, called the internal capsule. And the internal capsule runs out of the cerebrum down into the midbrain. So that's the cerebrum. There is some white matter. There's the gray matter, which is the cortex, and then some white matter. The cerebellum. Oh, so by the way, let's go back to the cerebrum. I mean, you can see it pointed out here, so it's not like I have to point to it, but all of this gray matter. Notice the sulci, which is the grooves, and the gyri, which is the bumps. So you have the sulci, which is the grooves, and the gyri, which is the bumps or the mounds. Deeper sulci are called fissures, like this lateral fissure right here and this transverse fissure right here. So deep sulci are fissures. So sulci and gyri, and then deep sulci are fissures. So that's the cerebrum. The cerebellum. Here's the cerebellum. The cerebellum is why you are coordinated, and we'll talk a lot about that. But the cerebellum, this is a coordination center of your brain. Now the diencephalon, you can see that right here. I know it's not pointing to it, but there it is right there. The diencephalon has 3 parts to it. The diencephalon has the epithalamus, the thalamus, and the hypothalamus. And that's all the diencephalon. And by the way, some people think your retina is part of that. I wouldn't disagree. Embryologically, the retina came from that area. Then you have the midbrain, and the midbrain contains the well, let me back up a little bit. These 3 parts right here, the midbrain, which is green, the pons, which is light blue. Well, actually, I'm sorry, that's not the pons. Here's the pons. The midbrain, which is green, and presumably a little of this light blue area. Talk about that. The pons and the medulla oblongata. These 3 parts, the midbrain, the pons, and the medulla oblongata, is collectively known as the brain stem. So the midbrain, pons, and medulla oblongata is known as the brain stem. The midbrain is your cerebral peduncles and your red nucleus of substantia nigra, your corpora quadrigemina, which is your superior colliculi and inferior colliculi. And even as I say this, it means nothing to you, but it will shortly. The pons is right here. The pons is involved in, it's basically a relay station. It's a relay station to the cerebellum. It's a relay station from the medulla oblongata to the cerebrum. But it does contain some respiratory centers. So the pons talks to the medulla for respiration. Otherwise, it's a relay station. And then the medulla oblongata, that does heart rate, blood pressure, respiration, heart rate, blood pressure, respiration. It does things like that. Some other autonomic functions. So that's the medulla. So those are the 6 regions of the brain. The brain weighs about 3 pounds. Now I say the brain weighs 3 pounds. The brain weighs 3 pounds if you were to put it on a scale. But it is almost as if it was swimming. It's floating in cerebral spinal fluid. So you know how when you go into a pool, if you were to stand on a scale in the pool, you wouldn't weigh as much as you do if you're standing on a scale outside the pool. In fact, you might float. Some people are really good floaters. Some people are not so good floaters. But the same thing with the brain. If you were to take the brain out and weigh it, or the brain adds this much weight to you, and that's 3 pounds. But if you were to weigh the brain within its cerebral spinal fluid, it's only a couple ounces because the brain floats in the cerebral spinal fluid. All right. The brain has a normal range of volume, and that range of volume you can see here is 750 to 2100 milliliters. That's a range of volume that is completely normal brain volume. That means someone with a 750 milliliter brain is just as smart as someone with a 2100 milliliter brain. Uh, there's not it's not saying that someone with a smaller brain is not as smart. We're not saying that at all. Okay, so that's an overview of the brain. This is taking out the diencephalon and the brainstem right here. Here we go. So the diencephalon, remember, is the thalamus, hypothalamus, and it's actually the epithalamus, which you can't see here. You can't see the epithalamus. The midbrain right here, here's your cerebral peduncles. Now you got to be careful because these are cerebellar peduncles. They go to the cerebellum off the back of the pons. So cerebral peduncles, they're relay stations, relays. These are cerebellar peduncles, and the words are close to each other. Cerebellar peduncles. These two bumps right here, and there's actually 4 of them because there's 2 on the other side that you can't see. These are called collectively the corpora quadrigemina. Corpora means body, corpora. Quad means 4, quadrigemina, corpora quadrigemina. That's the corpora quadrigemina. The 2 on top, because you can't see the one on the other side, is called the superior colliculi. And the 2 on the bottom are called the inferior colliculi. Remember, there's another one on the other side you can't see. It's inferior colliculi. The superior colliculi are involved in visual reflexes, and the inferior colliculi are involved in auditory reflexes. So from the eye, from the ear, and reflexes involved. That's the midbrain. We have some more things about the midbrain to talk about. We have to talk about the tegmentum and the red nucleus and the substantia nigra, but we'll wait till we get to the images of it. The pons is basically a relay station. There's vertical fibers and there's transverse fibers. The transverse fibers run back to the cerebellum. The vertical fibers run up and down from the medulla to the diencephalon and ultimately the cerebral cortex. So we got transverse and vertical fibers there. But it does also contain a respiratory center, which is very important. And then lastly, we got this medulla oblongata. The medulla oblongata involved in heart rate, blood pressure. That's the cardiovascular center, by the way. We can collectively say that those areas of the medulla oblongata are the cardiovascular center. Whoops, can't spell center, cardiovascular center. It's involved in respiration and some other things. So that's what they're involved in. All right, so that's a picture of the brain, and we're still just doing an overview, just so you know. Let me go back up here to the diencephalon briefly. 90% of all sensory input to the brain comes through the thalamus. 90%. So 10% of it doesn't. It bypasses the thalamus and goes directly to its cortical region in the cerebral cortex. But 90% of all sensory input comes through the thalamus. And the thalamus talks to the hypothalamus, which is big in autonomic regulation, and it's the connection between the brain and the endocrine system. The hypothalamus actually is the master of the master endocrine gland. The hypothalamus tells the pituitary what to do. Here's the pituitary. That's the master endocrine gland. And then the hypothalamus is nervous tissue, so it's not an endocrine gland, but it certainly does tell the pituitary what to do, and we'll talk more about that as we go. You can see how you can pull out this diencephalon and the brainstem out of, it's in here, it's underneath the cerebral cortex, it's within the brain, and of course that's going to become abundantly clear to you in lab, because we're going to take brains apart and learn all these structures. So that's how you take the brainstem and diencephalon out of the brain, and you can look at the parts of it. All right.
The brain is divided into 4 regions: the cerebrum and cerebellum, and the deeper-lying diencephalon and brainstem. The cerebrum consists of 2 large hemispheres, whose surfaces are distinguished by thick folds called gyri. that are separated by grooves called sulci. Each cerebral hemisphere is composed of 4 lobes. In a lateral view, the frontal lobe, temporal lobe, parietal lobe, and occipital lobe can be identified. Each lobe is responsible for specific functions. Overall, the cerebrum is considered the seat of intelligence, responsible for information processing, language, personality, and conscience. The cerebellum is composed of hemispheres with many folds. Its primary function is to coordinate motor activities. Structures covered by the cerebral and cerebellar hemispheres can be seen best in a mid-sagittal section. The diencephalon is composed of the thalamus, hypothalamus, and epithalamus. The thalamus is a major processing and relay center for information entering the cerebral hemispheres. Ventral to the thalamus is an area called the hypothalamus. It has a primary role in homeostasis, including regulation of body temperature, food and water intake, sleep and circadian rhythms, emotional responses, and memory. The epithalamus is dorsal to the thalamus and has 2 parts. The pineal gland is an endocrine organ, while the habenula is a relay center between the midbrain and limbic system. The brainstem is a vertically oriented stalk located between the diencephalon and the spinal cord. It can be divided into the midbrain, the pons, and the medulla oblongata. The midbrain is the most rostral segment of the brainstem and has an important role in visual and auditory reflexes. The pons is located caudal to the midbrain and is characterized by a prominent anterior bulge. It contains numerous ascending or descending fiber tracts. The cerebellar hemispheres are attached to the pons. The medulla oblongata extends from the pons to the foramen magnum, where it is continuous with the spinal cord. All nerve fibers traveling between the brain and spinal cord pass through the medulla oblongata. It also contains nuclei concerned with basic physiological functions, such as respiration, blood pressure, and heartbeat.
Now, she mentioned nuclei in that video. I want to remind you what a nuclei is. In the nervous system, because we're not talking about protons and neutrons here, we're not talking about a nucleus of an atom. Nuclei in the nervous system are a collection of soma. Now, nuclei specifically are a collection of soma in the central nervous system. Because if you have a collection of soma in the peripheral nervous system, as you already know, we call it a ganglia, ganglia is plural. We call them ganglia. We call one of them a ganglion. And the most famous ganglia you've learned so far in lab has been the sympathetic chain ganglia. You know, the sympathetic chain ganglia are the pair of chain ganglia on each side of the spinal cord that have the white ramus communicans going into it and the gray ramus communicans coming out of it. So you already know what ganglia are. They're a collection of soma, cell bodies of neurons in the peripheral nervous system. So ganglia are peripheral nervous system, and nuclei are central nervous system collection of soma. So just to remind you, because she says there's nuclei in the medulla involved in regulatory events, and that's true, and we'll talk about them. But just remember what a nuclei and a ganglia is. All right, now ventricles. Within your brain, you have these cavities or spaces called ventricles. And this one that looks, to me, it kind of looks like a jaguar. Right there, kind of like a jaguar. I don't know. I mean, when I say jaguar, not the real cat. You know the emblem on my car. You know my Jaguar car I have out in the parking lot. Well, on the front of my Jaguar, I have that statue on the front. That's kind of looks like a jaguar to me anyway. Those are the lateral ventricles. So here's one lateral ventricle. Here's the other lateral ventricle. One of them is numbered number one, and one of them is numbered number two. I forget which one's which. It doesn't really matter. So these are lateral ventricles here. And by the way, within all of these ventricles, cerebral spinal fluid is made. It's produced here. But after the cerebral spinal fluid is in the lateral ventricle, it doesn't stay there. It flows into this third ventricle right here. And right here where it flows into the third ventricle, here and here, it flows through this interventricular foramen. Foramen means hole. So there's a hole connecting the lateral ventricle to the third ventricle, or a canal, if you will. And the cerebral spinal fluid flows from the lateral to the third, from this lateral to the third, which makes this the third ventricle, right here and here. Now this right here, I mean, it looks like there's a hole in the ventricle, but remember, the ventricles are holes or cavities or spaces. The ventricles themselves are spaces. So this right here, what I'm circling, is actually solid tissue. And it's called the interthalamic adhesion. The thalamus is actually two lobes, the right lobe and the left lobe. And this interthalamic adhesion connects the two lobes of the thalamus. And the third ventricle is around that interthalamic adhesion, or if you will, between the two lobes of the thalamus. And we'll look at that again in the lab. It's hard to explain it right here. This is the third ventricle. Third ventricle makes more cerebral spinal fluid, but it also receives CSF from the lateral ventricles. And then the third ventricle, the CSF made there, can flow into the fourth ventricle, and it does that through this cerebral aqueduct right here, from the third to the fourth. Cerebral aqueduct, CSF flows from the third to the fourth through the cerebral aqueduct. All right, now from the fourth ventricle, I have apertures. I have two lateral apertures and one median aperture. It's out of these three holes that the CSF flows to where we really need it, and that is into the subarachnoid space. So the cerebral spinal fluid flows out of the fourth ventricle into the subarachnoid space via these three apertures of the fourth ventricle. And those three apertures are called the lateral aperture here, the lateral aperture here. There's two lateral apertures. And the median aperture here. And now the CSF is in the subarachnoid space. So these are the ventricles of the brain. All right, and we'll study that some more in lab. The cerebral spinal fluid, here's the cool thing. We all make about half a liter of cerebral spinal fluid today. That's 500 mils. But if I were to measure your cerebral spinal fluid volume right now, you'd probably only have about 150 milliliters of it. So how can it be true that you make 500 milliliters a day, but at any given time you only have 150 to 180 milliliters? And the answer is, well, it must be reabsorbed. And it is reabsorbed. And I will show you where it's reabsorbed. In fact, it takes about 8 hours to turn over all your cerebral spinal fluid. So you're constantly making new, and you're constantly reabsorbing old, and in about 8 hours you overturn all of your cerebral spinal fluid. And that should make sense to you, because how many times does 8 hours go into 24? And that's 3 times. And if you have 150 milliliters at any given time, how many times does that go into 500 milliliters? Well, if you do 3 times, that's 450, I believe. So that's almost 500. So about 8 hour turnover. All right, so that's the ventricles, and we'll look at the circulation and reabsorption in a second. Now, embryologically, this is what your brain looks like. Embryologically, in about 3 weeks, you have 3 brain vesicles. They're called primary vesicles. This is going to become your brain. It's not your brain yet. It's going to become your brain. These 3 vesicles are the prosencephalon, mesencephalon, and rhombencephalon. The mesencephalon's name does not change. So the mesencephalon is easy. The midbrain is the mesencephalon. Matter of fact, even in the adult brain, we sometimes call the midbrain the mesencephalon. But the prosencephalon does change. About 6 weeks later, the prosencephalon differentiates into 2 secondary vesicles. And those are the telencephalon and the diencephalon. The telencephalon becomes your cerebrum, your cerebral cortex, and also the lateral ventricles. The diencephalon becomes what we talk about, the the epithalamus, the thalamus, the hypothalamus. That's the diencephalon. And we still use the word diencephalon even in the adult brain to describe those three thalamic regions, the epithalamus, thalamus, and hypothalamus. And in addition, because the third ventricle is between the two lobes of the thalamus, the third ventricle also comes from the diencephalon. Now, remember back when I was doing the diencephalon about 5 seconds ago, I said that some people think the retina of the eye is part of the diencephalon. Well, I wouldn't disagree with that. Uh, the retina of the eye is a product of the diencephalon. So uh, a lot of books treat it that way. All right, the mesencephalon came from the primary vesicle, the mesencephalon. Its name doesn't change, but what does the mesencephalon become? The midbrain. And because behind the midbrain there's the cerebral aqueduct between the third and fourth ventricles, the cerebral aqueduct also comes from it. Remember what the midbrain is: the cerebral peduncles, the corpora quadrigemina, which are really the superior and inferior colliculi. So the mesencephalon doesn't change, other than to mature and develop it, but its name doesn't change and it becomes its mature brain feature. The rhomencephalon, however, does. It becomes two other things. Similar to the prosencephalon, which differentiates into the telencephalon and diencephalon, the rhomencephalon differentiates into two things: the metencephalon and the myelencephalon. Now, the metencephalon is the cerebellum and pons. I always remember this because the cerebellum is hooked to the rest of the brain structures via the pons. The pons contains the cerebellar peduncles. The pons has transverse fibers that connect the rest of the brain to the cerebellum. So the metencephalon becomes the cerebellum and pons, and the myelencephalon becomes the medulla oblongata. So these are embryological brain regions, the primary vesicles at about 3 weeks, and the secondary vesicles at about 6 weeks. And these are the adult brain structures. Well, I mean, you have them when you're born, and your brain still has a lot of development to go through, but we'll call them adult brain structures. So there you go. That's the embryonic brain and how it becomes the adult brain.